So we're going to do disease. Let's have a look at the syllabus. So yeah, it's communicable disease, diseases that spread due to pathogens, a disease-causing microorganism. And you need to be aware of non-specific defence, as in how we stop microbes getting into our body. So yeah, our skin's the first one that's in the syllabus, and this is just these layers of overlapping cells. So there's layers of overlapping cells, and they're dead. There's a dead outer surface. So there's nothing like no food for them if it's dead cells. Nose has got hairs and mucus. As you can see, mucus traps microbes and kills them as well. And it also, yeah, kills bacteria. It's got a bacteria killing chemical in it. The hairs just like increase the surface area. So there's like a web of hairs covered in mucus so it can filter the air as it comes in, filters the air using the hair. Tricky. Just be careful with trachea and bronchi. It is also the bronchioles, but for some reason bronchioles are not on the syllabus. But yeah, we have lining our tubes that carry air into our lungs. So there's a tube. These are the cells that are lining it. There's a slightly different one there. Another one, and these have little fine hairs called cilia. Yeah, so these are microscopic hairs. Hairs, which equals cilia, one L in cilia. And there's also mucus. So this is a cell that's making mucus. So it's similar to the nose. There's a sticky surface and hairs to trap and remove them. But yeah, the cilia are more for removing the microbes. But yeah, plus there's mucus as well. It's the same mucus as in the nose. Yeah, so just be careful with cilia. There's not cilia in the nose. Cilia are microscopic, they're found in the lungs. A lot of people say throat. Yeah, the throat is just the back of the mouth area where you can decide to go down the esophagus or you can decide to go down the trachea. And stomach, as you know, it makes HCl, hydrochloric acid. Plus there's also protease enzymes. But focus on the HCl, it's therefore a pH2, and that kills most pathogens. Kills most pathogens, unless you're protected by food. So that's our non-specific defence. And then it says students should be able to explain the role of the immune system. So we've got phagocytosis, antibody production, antitoxin production, and then it goes into vaccination. Just before we do that, I thought we'd just do a summary of the different diseases. I've not put the syllabus here because quite frankly, the syllabus is very detailed notes. So this is just a very quick overview of all the ones it says in the syllabus. So the disease on the left-hand side, so the first one let's do is AIDS. That's viral, well, it's caused by a virus. How it's spread, it's either injecting needles or unprotected sex. So I'm just going to put needles, stroke sex. So it's drug users who shared needles. It's unprotected sex. Symptoms, it starts with a fever, but it progresses to loss of immune system. So you have lots of other infections. And that's what AIDS stands for. It's acquired immune deficiency syndrome. The virus is called HIV. So HIV isn't the disease. HIV is the virus that causes the disease of AIDS. Prevention is like clean needles, but in terms of what you've been doing in year 11, it's talk about the barrier method of contraception. Yeah, you need to stop fluid transfer. Yeah, how it spreads. Yeah, so it's all to do with fluid transfer. Measles is also viral. It's a virus. How it spread. This is droplets breathed in. So it's the droplet one. So coughs and sneezes, etc. Symptoms: you get a fever, rash, and prevention. It's vaccination. So the news at the moment is measles outbreak. I think in Birmingham. It's also quite prevalent in London area as well. So yeah, vaccination. It's the MMR vaccine: measles, mumps, and rubella. Rubella is German measles. Then we get to the two bacterial diseases. So these are bacterial. And often they give like a tick box question in an exam. It's a low level question. So yes, both these are going to be bacterial. Gonorrhea. This is unprotected sex again. So sex, unprotected. Symptoms. It's like a green yellow discharge. So it's discharge from penis stroke vagina, and painful urination. Painful urination. Again, so it's all squashed down a lot, but I'm trying to keep it on one page. Prevention, again, it's the barrier method. I've missed off treatment on here. I'll come back and do treatment at the end. I have to squeeze it on somewhere. Sorry about that. So that's barrier method of contraception. So same as before. Salmonella is it's undercooked poultry. So it's poultry badly cooked, as in like chicken. Symptoms again: fever, abdominal pain, diarrhea. Prevention: it's all food hygiene. Plus we vaccinate chickens. Malaria is a protist disease. It's still microscopic, but it has a nucleus. Bacteria don't have a nucleus. It's spread by a mosquito. That's why I spell it right. And that's called a vector, as in it's a spreader of it. So mosquitoes, you get fever on that one. Prevention, it's sleeping under nets. Here's a good example. So all these are the human ones. Just in terms of treatment, let's just I'll pop it kind of over on this side. For AIDS, well, to stop it getting to AIDS, use a antiretroviral drugs. Again, it does say this in the syllabus. Antiretroviral drugs. So HIV is a retrovirus, which means it kind of converts its genetic material into DNA within the cell. So it's an antiretroviral drug. So it's a drug you have to take every day to stop the virus from replicating inside the cells. Measles, there's no treatment apart from intensive care. So it's deadly as measles. So it kills like 1 in 4,000 in the UK. So miles more miles more dangerous than like COVID was in terms of killing people, and it affects the young more than the old as well. Gonorrhea is antibiotics. It's a bacterial disease, so you can use antibiotics. You may also use them for salmonella, but unlikely. You'll just get better from salmonella. You just have to treat the symptoms, but you should be okay. Treating malaria, again, there are anti-malarial drugs. As far as you're concerned, it's just knowing that antibiotics are only useful for bacteria. You can't use them against viruses. Let's do the other two quickly. Rose black spot. Again, these are plant diseases. I'm going to honorary change to a green colour just for fun. There we go. Type of pathogen. So rose black spot is our fungal disease. So it's a fungus. How's it spread? Well, it tends to be like the fungus is on the leaf, and then it'll rain on the leaf and splash the fungus onto other leaves. So it splashes. Splash from rain. So if you're in the same vicinity, you'll get infected. The symptoms on the leaf, you get black spots. Like the leaves kind of go yellow as well, and there's no really preventing infection. You have to just get rid of plants that have it. Yes, so you don't try and treat the plant. You just destroy infected plants. So you burn them. That's why you often see like gardeners having like a little fire. They might be burning some infected plants. And TMV is tobacco mosaic virus. Um, Again, TMV, HIV, it's virus. How's it spread? This can be, so this is a bit like malaria, but it's more an aphid vector. The green fly is an aphid. So again, it tends to be the aphid feeds on one plant and then goes onto another plant and injects the virus in. Symptoms, you get a mosaic of like less green. So you get it's like loss of chlorophyll, but like patchy. Because chlorosis is also loss of chlorophyll, but that's down to lack of magnesium ions. And prevention, again, you'll just destroy it by burning. So yeah, a bit messy. Make your own little table. Would be my tip. But yeah, if you look in the actual syllabus, there's loads of detail there. This might be a two-parter because I've already done 12 minutes and I've got probably about another 10 minutes left. So you might want to like do half if you're trying to do things in little bits. So just to remind you about pathogens, these are disease-causing. These are the main two for us: viruses and bacteria. And viruses, these invade cells, and they destroy cells. Whereas bacteria, these make toxins that make us ill. And then you need to know about protection from disease. So this is our phagocyte. All these cells are white blood cells. So that's our phagocyte. This is a virus or a pathogen. This is the phagocyte engulfing. So I put right in the exam, engulfs the pathogen, and then it digests the pathogen. Engulfs and digests, the pathogen. So these patrol our body. If they bump into a virus or a bacterium or a protist, they will engulf and digest it. Okay, so that's a phagocyte. 2 marks, they engulf and digest. This is the other class of cells. This is a lymphocyte. We do these together because that's also a lymphocyte below it. These are antibodies. This is our pathogen again. Could be a virus. Here you can see that the antibodies bind to antigens. This is when it's tricky on the pathogen. And then this causes, the pathogen is destroyed. Now, we're missing lots of detail here because it's not on GCSE, but in effect, it speeds up the phagocyte process. If you're covered in antibodies, the phagocyte will do this way, way faster. It doesn't need to like decide if to kill you or not. You're already like highlighted for destruction. So the little triangles on the pathogen are called antigens. Antigen is a portmanteau. It's a squashy together of, it means antibody generator. So when our body gets exposed to an antigen, our body makes antibodies against it. So apologies for the antibodies, antigens, and before we have antibiotic, there's 3 anti-words that are all confusing. But make sure you know each one. And then number 3, here's another lymphocyte, but now it's making antitoxins. Still look exactly the same. They are a kind of antibody. This is a bacterium, a pathogen. Then these are now the toxins. And here the antibodies bind to toxins. Oh, sorry, antitoxins, sorry. Bind to toxins. And the toxin is neutralized or destroyed. Don't put killed. You can't kill something that's not alive. So we have a way of dealing with, again, there's another way of dealing with viruses inside cells as well, by another kind of lymphocyte, but it's not on the syllabus. So yeah, so make sure you know the role of lymphocytes and phagocytes. And then lastly, I thought we'd just zip through immunity. So here I've got a graph, it shows time in days. So I've got 7 days shown here, and then I've got another 7-day period, 10 days later, sorry, 10 years later. And this is the first exposure to an antigen. It could be an antigen on a virus or a bacterium, or it could be part of a vaccination. And then this is the second exposure to the same antigen. Okay, so we're going to go for like a 7-day period of being infected once and then infected again. And then on the y-axis, I've got antibody concentration in the blood. So let's assume I'm going to get flu. I get exposed to the flu virus on day 0. Well, it takes me about 3 days to make antibodies. So number 1, you see there's number 1. It takes time. It takes like 3 days, in this example, for the body to make antibodies. Therefore, we get ill. Number 2, antibodies are made by what kind of cell? That's what last year in a GCSE exam, by lymphocytes. And 3, the antibodies are used up. Antibodies, so they get destroyed in the process. Antibodies are used up or don't last long. Okay. However, the second time I get exposed to it, and it might be next week, next month, or here it's 10 years later, number 4, you can see that the delay is hardly anything. Yeah, it's not long enough for me to get ill. So we now have a faster production of antibodies. It is due to memory cells. It's a memory lymphocyte, but you don't need to know memory cells. It's not on the syllabus. So basically, when I go through it the first time, I make many memory cells, and they recognise it faster. So faster production of antibodies, and more antibodies are made, or a higher concentration. So yeah, pathogen is destroyed before symptoms. So you never get ill. You might just have like a swollen lymph node or something. And crucially, number 5, which I'll put here, 5, yeah, the antibodies last for a long time. Antibodies last for months to give immediate protection against whatever this antigen is attached to. So in terms of vaccination, the aim of vaccination is to, and we'll just put it down here for vaccination. So if this is a vaccination, our syllabus talks about we just inject a weakened version of pathogen. So it can't cause disease, but it has the same antigens. SMA, I can't spell same. Same antigens as pathogen. So that just makes us go through this immune response over here. It's called the primary immune response. So yeah, you recognize the antigens and you make these things called memory cells. So when you get exposed to the pathogen properly, like the measles virus that's roaming around the country, if you're watching this in 2024, then you'll make antibodies faster. You make them so fast you don't get ill, and therefore you are classed as immune. The only other word is called herd immunity. That's when 95% of the population are immune. Therefore, the 5% are protected. So not everyone can have a vaccine. not everyone gets immunity from a vaccine, um, and yeah, some people have medical conditions where it's not safe to give them a vaccination. So that's what herd immunity is, and that's yeah, the target is to get to 95%. So it's a bit messy at the top, but hopefully um, there's some useful stuff there. Again, use the syllabus, and that's it. 20 minutes. Perfect.